suggests they are called non-histone chromosomal proteins, NH and C. Okay, NHC proteins. I told you that during nucleosome formation, there are histone proteins, right?
Yes, sir.
Histone proteins are used during the early, the early or initial levels of condensing of the DNA. So we need to compact and condense the DNA in order to keep it inside a very small nucleus, right?
Yes, sir.
So for that, for the initial levels, to make a nucleosome or a bead on a string structure, we need histone proteins. Now they are of various kinds. Histone octamer containing H2, H3, and H4, and H1 is there in the outside of the histone octamer. There is H1 histone. So these all are histone proteins. As we go forward, then we used NHC, non-histone chromosomal proteins, to pack it further, okay, into a chromosome. So chromosome is a very, very densely packed and compact form of DNA. Okay, so that's all you have to know about the NHC proteins, that they are used or they are required in high order packaging of the DNA. Okay.
Yes, sir. Thank you very much.
Okay.
Good evening, sir.
Good evening, Arpit. How are you?
Sir, I am good. How are you?
I am very well. So today I might not be able to use the video, so we'll go with the audio only. Okay. So please be discussive so that I know if I am audible to all of you.
Yes, sir.
Okay. Okay. So let me go to Samara today. Samara, are you there? Can you give us a brief summary of the last class, the introduction of the chapter?
Yes. So first we started with DNA. It is a deoxyribonucleic acid. Deoxyribo means deoxyribose, that is absence of ribose. That is a pentose sugar. It has 5 carbon. Nucleic means it is present in the nucleus, and it is a acidic molecule. Then DNA consists of, it has 3 components, that is the phosphate group, pentose group, and nitrogenous base. Pentose group, there are DNA and RNA, and in the nitrogenous base there is adenine, thymine, or uracil and RNA, then guanine and cytosine. Then nucleotide. Nucleotide consists of a phosphate group, sugar, and base, and nucleoside only consists of the.
What is nucleotide?
What's that?
What is nucleotide?
Yeah, nucleotide. It consists of phosphate, carb, sugar base, pentose sugar, and
what it is. I'm not asking what it consists of. What it is.
It is a polymer.
No. DNA is a polymer.
Monomer.
Yes, nucleotide is the monomer of DNA. Okay.
Okay, sir.
Yeah.
Then 2 nucleotides they are attached together through phosphodiester linkage to form a dinucleotide. Then in RNA, every nucleotide has an additional hydroxyl group, that is OH group, at the second position in the ribose. Then we discussed about the structure of DNA. It is a double helix structure. Frederick in 1969 discovered that DNA as an acidic substance is present in the nucleus. He named it nuclein. Then in 1953, James Watson and Francis Crick gave the double helix structure of DNA based on the experiments done by Rosalind Franklin and Morris Welskin. They did X-ray diffraction experiment on DNA. Then we discussed a question about give the salient features of double helix structure of DNA. So the first point it is that it is made up of 2 polynucleotide chains. The backbone is composed of the phosphate and the sugar group, and the bases it project inside. Then the second point is both polypeptide chains of DNA have anti-parallel polarity, that is 5' to 3', then 3' to 5'. Then the third point is the nitrogenous bases, they are paired through hydrogen bonding. Adenine forms 2 hydrogen bond with thysin. Then guanine forms 3 hydrogen bond with cytosine. They are always purine binds with pyrimidine. This specific binding helps to keep the distance between the 2 strands uniform. Then the fourth point is the 2 chains are coiled in a right-handed fashion, that is clockwise. One turn is called a pitch. And fifth point, the plane of one base pair, it stacks over the other in a double helix manner. The stacking along with hydrogen bonding provides it provides stability to the helical structure. Then we discuss about central dogma in biology. The central dogma, it is a molecular biology. It defines the flow of genetic information in a living system. It always is in a unidirectional manner from DNA to RNA, then RNA to DNA. So replication takes place.
Not RNA to DNA. It's DNA to RNA and RNA to proteins.
Yes, DNA to RNA.
DNA is an exception.
Environmental.
Yeah. Yes.
Then we discuss about packaging of DNA helix. The pitch of DNA is about 3.4 nanometer. The distance between the 2 adjacent base pairs is 0.34 nanometer. Then one strand is of 3.3 nanometer length. Then one pitch approximately consists of 10 base pairs. Then the total length of DNA in a diploid mammalian cell is approximately 2 meter per cell. DNA, it is a negative charge. It is negatively charged. It
binds around.
Yeah, it binds around the histone molecule. 8 proteins combine to form histone.
No, all of the 8 are histones. So 8 histone proteins combine to form histone octamer.
Yes. Then this is called a nucleosome. Then
what happens around the nucleosome? DNA binds. This black thread is the DNA. Can you see that?
Yes, sir.
Yeah.
And there's also H1 histone protein, which clips it. Every nucleosome has 200 base pairs of DNA. And
then we finished with the Nhc proteins.
Yeah, beyond the level of chromatin fiber, the packaging of DNA at higher orders requires additional protein, that is Nhc protein, that is a non-histone chromosomal protein.
Yes, so the name suggests they are chromosomal proteins. So they play a role in making chromosome. Chromosome are the highest order of DNA packing, where the DNA thread finally becomes a chromosome. Okay. And I ask you to go back and read about chromatin, heterochromatin, and euchromatin. So did you do that?
Yes, sir.
Yes. So what is a heterochromatin and what is a euchromatin?
Heterochromatin. I just like learned saw the features difference between them.
Sorry, come again. What is the difference between the two?
Yes, sir. Sir, the heterochromatin is highly packed, and sir, the euchromatin is loosely packed as compared to. Sir, the genes in the heterochromatin are inactive, and in euchromatin they are active. Sir, and heterochromatin are present in the nuclear envelope, and sir, the euchromatin, it occupies most of the area of the nucleus. And
yes, the third point actually is it's not required at your level because it's not entirely true always. So you just have to understand that when we are compacting DNA, okay, now everyone pay attention to what I'm saying, okay? So when we are packing DNA into more and more compact form. Okay, finally to make a chromosome. So this is basically how a chromosome looks. We have already studied. Let's first write down the heading. So we are going to read now two different types of chromatin. Euchromatin and heterochromatin. Now the word "eu" in biology always means true. Bacteria, euchromatin, eukaryotes, everything. So this is the true chromatin. By true chromatin, I mean that it is loosely packed. And when it is loosely packed versus heterochromatin, which is tightly packed. So this packaging actually defines that if it is loosely packed, then the DNA segment, which is euchromatin, which is loosely packed, it will be easier to access by the proteins. So the proteins that are accessing the DNA for transcription to make RNA out of it, which will eventually help in making proteins in the cell. So that part of the DNA is accessible by the proteins. Yes or no? If you understand it this way, so if I ask you to move through two different kinds of forest patch, one is a bamboo forest patch. You know, bamboos, they grow very, very close together and in a very dense manner. And the other bamboo is like the other forest is of any other tree which grows at a distance from each other. So through which of the patch it will be easier for you to move?
Bamboo.
It will be easier for you to move through bamboo?
Sorry, the normal one.
Yeah, because bamboo is very densely packed. You might not be able to, the space provided between two bamboo will not be enough for you to move through it, right? So you will find it difficult. And if I send, so you are like a dye, you are like a stain. So if I send a stain to color that part of the DNA, so which part, heterochromatin or euchromatin, will allow the stain to pass through it, and which one will retain the stain?
Euchromatin will allow it to pass through it, and heterochromatin will actually stain.
Yes. So that's why euchromatin is lightly stained. When you stain it with a dye, it's lightly stained because the stain passes through it, whereas heterochromatin is dark stained. So this way you can actually make a chromosomal map of the region which is euchromatin and which is heterochromatin. So let's say if this is a chromosome, right? This is how you see a chromosome, right? In general. So
yes,
if
sir.
we use the same color, so some regions of this chromosome, sorry, when you will send the stain, it will stain it dark. Okay, some regions will stain dark. You understand? Let's say these are the dark stained regions. And with the same stain, other regions will be lightly stained. Do you understand, everyone? Yes, no, something?
Yes, sir.
So the region that you are seeing here as darkly stained. Okay. This part here, this is a heterochromatin. Okay, because it's dark stained. And also heterochromatin is transcriptionally inactive. Because obviously it makes sense, right? Why is it inactive? Arpit say the sentence that the genes are inactive. Why? Why are the genes inactive in heterochromatin region? Whereas in this region, which is lightly stained, which is euchromatin, whatever genes are present here, they are transcriptionally active. Can anyone tell me why? I already gave you the answer in the beginning. Arpit. Why do you think genes in this heterochromatin region are inactive and genes in this euchromatin region are active? Am I audible? Am I audible, people?
Yes, sir, you are.
Okay, so no one is speaking on purpose.
Because in heterochromatin they are densely packed.
So what is the problem with the densely packed region of the nucleus? Why the genes can't be active in that region? Let's go on, complete the answer. I think you are thinking along the right path. How do we, how is the
Because
gene?
they can't access the transcription.
The proteins that are important, the factors, the molecules that actually make RNA from a DNA segment cannot access that region of the DNA. That's why. You understand?
Understood, sir.
Yes. So if it is densely packed, enzymes like RNA polymerase, which makes RNA from DNA, cannot reach that region. It is so much packed and folded that that enzyme cannot reach that region to be able to open the two strands of the DNA, get inside the two strands and make a RNA out of one strand. Okay, that's not happening. So that's why genes in this region are inactive, whereas genes in the other euchromatin region are active. They can be actively transcribed. That's why I have written transcriptionally inactive and transcriptionally active. Okay.
So,
yes, Arpit.
Can you explain a little again, sir? I got stuck into some position.
Yeah, so I was just asking that why do you think the genes present in the heterochromatin region are transcriptionally inactive, and why are the genes which are present in euchromatin region transcriptionally active? So can you think of a reason, Arpit?
So because like they are loosely packed.
Yes, so because they are densely packed, heterochromatin is densely packed. Factors which are important for transcription, the enzymes like RNA polymerase, it cannot reach that gene. Okay, and cannot transcribe that gene into a RNA. So that's why they are transcriptionally active. Here it's loosely packed, so things can reach and the machinery can reach, about which we will study in today's class, and can transcribe. You know, and this is a very good way. If you think, have you ever thought that every organism, including humans, we are basically produced from one single cell, which is called zygote?
Yes, sir.
And every other organism, elephant, birds, reptiles, cats, dogs, all are produced from one single cell, which is called a zygote. Yes. Yes. You understand? Am I audible?
Yes, sir.
Yep. So how does, as we develop one zygote, how does one zygote give rise to so different kinds of cells, functionally and structurally both? So a zygote is just one single cell. And when we take birth, We have neurons, muscle cells, skin cells, eye cells, photoreceptors, which are present in the eye. We have fat cells, all different types of cells of different organs, which look entirely different, and also their function is also different. So how do you think when we start with one zygote having the same genetic material? So ultimately, all our cells in the body have the same genetic material, have the same number of genes, right?
Yes, yeah.
So the information present in each and every cell in our body is same. But still, how does each and every cell of our body can have different function and structure? Have you ever thought about it? Many genes which are required, now this is the answer, many genes which are required only in a particular organ or a particular type of cell is expressed in that cell type. It is not expressed in other cell types because by using this mechanism, those genes which are not required in a particular cell are heterochromatized. Okay. You make those genes transcriptionally inactive so that, let's say there is a gene X, and that gene is responsible for some structural feature of a neuron. So if that gene expresses, the stem cell will develop into a neuron, let's say. But you have to make bone cells or you have to make blood cells, not neurons. So you will not like that gene to be expressed, right? You will like some other sets of the genes to be expressed. Yes or no?
Yes, sir.
Some genes are common, which are expressed in all cell types. Some genes are specific. So these specific genes make all the difference. And the way our cells regulate it is through this heterochromatin and euchromatin mode. This is one of the major ways. You understand? That's why we can achieve different cell types coming from one single zygote. Okay, everyone?
Yes, sir.
Just for you to, so this is, you have never thought of this question actually on your own? I used to think about it. I also, and there's one more question which is even more intriguing. How does the zygote knows that I am a zygote of a elephant? So I do not have to develop into a human, but I have to develop into an elephant or a chimpanzee or a bird, not in something else. It's a single cell, right? So you will say that it has different genes. Yes or no?
Yes, sir.
Yeah. But if I tell you, I asked you to go back when we were studying reproduction. And if you look at the early embryo, early embryo of every organism looks almost the same. So it has a head bud and it has a tail bud and it has limb buds like this. And then there is this kind of thing. So every embryo looks like this during the early development, something like this. It also has gill slits. Human embryos have gills in the beginning. We have gill slits, not functional gills. But at the region where fish have gills, our embryo also starts developing gill slits. In fact, that is one characteristic feature of chordates. Class 11th, Animal Kingdom. If you have read it properly, you have read about chordates, right? Do you remember anything or everything is blank? I'm not able to hear anyone if anyone is speaking. Are you also facing internet issues, people? Do you remember the features of chordates? No? Yes? Okay. Okay, so I think everyone is having some issues. So there you know that tail bud. So this rudimentary tail, gill slits, pharyngeal gill slits, which we call, these are two features of every chordate embryo. Even humans who do not have gills but develop lungs, in the beginning, the embryo starts developing gill slits, which later on do not develop. Okay. So that is why is that? Why do we need a tail bud and gill slits? Neither we have tails nor we have gills. So that's another interesting thing. If you want to figure about it, do read. You can discuss it with me if you come up with some information of your own. Okay. Now in today's class, you understood chromatin, euchromatin, and heterochromatin. So I'll start now. How did we actually figure out that the genetic material is DNA? So we studied everything about DNA, the packing, the structure, the features. We studied things. We now know about the chromatin, euchromatin, and heterochromatin. Now let's try to figure out how was genetic material found. Okay. So what is genetic material in our cell? Now people knew that there is something called a nucleus. Okay. And very early, there was I told you about Frederick Mischer, who also said that there is something in the nucleus called nuclein, which is acidic in nature. Yes?
Yes, sir.
But no one knew that this genetic material, which is present inside the nucleus, which Frederick Mischer is calling nuclein, what is it? What is its chemical nature? Is it a protein? Is it a lipid molecule? Is it any kind of a nucleic acid molecule? What is it? Is it a sugar? Is it carbohydrate? What is the nature, biochemical nature? Now, this hunt started very early with this one person called Frederick Griffith. So I'll start with the first thing, which is called the transforming principle. Transforming principle. Now, experiment. So we are going to read about some series of experiments done. So this transforming principle experiment was done around 1928 or 26. Not just check it out. It's 1928 or 26. One of it. It was done by Frederick Griffith. Okay. Now what Frederick Griffith did was, he was the first one to figure out that something can be transferred from one organism to another or one cell to another, which can lead to change in the physical form of the other cell. Now this was very interesting because before this, everyone used to believe that genes can only be transferred vertically, like through generations, from parents to the offsprings, right? This is what we also studied in the Mendel in the 5th chapter. Yes, genes can be transferred from the parent generation to the offspring generation. Yes or no?
Yes.
But here, the first time it was shown, so he used the model system that he used for his experiment.
So
Was
can you
strepto.
from the start?
Yes, Arpit, tell me.
Can you explain from the beginning?
This is the beginning.
Okay.
Yes, we are talking about the first experiment, which is called transforming principle experiment, which was done by Frederick Griffith in this year, around this year. You can confirm the year. Okay. Now, Frederick Griffith, sorry?
The year is correct, sir.
Okay. Thank you. So Frederick Griffith used the bacteria called Streptococcus pneumoniae. And you know about this bacteria, right? Where have you heard the name of this bacteria? It causes pneumonia. Okay. That's why it's called Streptococcus pneumoniae. So the infection, pneumonia infection that happens in the lungs is caused by this bacteria. So this bacteria has two types of strains. So the first strain, strain 1, is known as smooth strain or S strain, okay? And the strain 2 is called rough strain. Strain means types of bacteria, okay? Or it is called R strain. Is it clear, everyone?
Yes, sir.
Yeah. Now, this smooth strain is actually virulent or lethal. Like this kills. Whereas the R strain is non-virulent or non-lethal. It causes the infection, but it is not deadly. And why is this strain deadly? Because it secretes a mucus coat around itself. This mucus coat is made up of polysaccharides. Okay. So this thing provides the virulence or the lethality. Whereas strain 2 does not have this mucus coat around. So that's why it does not look smooth. So when you culture these two bacteria, you will see that the first strain will form very smooth and shiny colonies. Is it clear?
Yes, sir.
So you can write it forms smooth and shiny colonies on a culture plate. So we culture bacteria on Petri plates, which are called culture plate. So on culture plates, they form smooth colonies and they form rough or non-shiny colonies. Because that shine and smoothness comes because of this coat, mucus coat around. Is this clear about the two strains, everything?
Yes, sir.
So this thing is not present here. Now let's see what is the experiment. So this is the experimental paradigm.
Sir, the line you wrote in black just now, this one, does it mean like from smooth it becomes rough?
This line?
Yeah. No, both of them.
This one?
Form smooth and shiny colonies on a culture plate, and they form rough or non-shiny colonies. These two.
Yes, because our strain does not have a mucus coat around. So when you culture them on a Petri plate, they form non-shiny colonies on a culture plate. It means nothing. I'm just telling you the properties of these two strains. Okay, this is just a bullet point. Okay.
Yes, sir.
There is a division in between. It's like difference between the two. Okay.
Yes, sir.
Now I'm coming to the experiment, the experimental paradigm. So the experiment, what he did, he took a S strain. Okay. Took the bacteria from the S strain and injected those bacteria in mice. So what do you expect? Based on the properties of the S strain that you have written above, what do you expect to happen? Mice will now get a pneumonia infection?
Yes.
And what will happen?
It will die.
Yes. So the mice die. It should die. because infection with the S strain causes lethality. We know that. Now, second, he took a R strain bacteria, did the same, injected into mice. And what do you expect now?
The mice will live
Why? Mice will get
because
infected
it's
or
not
not?
lethal.
Because it's non-violent.
Yes, but it will get infected, right? But
Yes,
mice
sir.
will live because they will overcome the infection and they live, because this strain is not that lethal. Now, the third thing that he did was, he thought, let me heat kill the S strain. So he used S strain. You know what is heat killing? Heat killing is when you boil or heat the bacteria die or become inactive. That's why we are told to boil and cool the water before drinking, especially those who are ill or those who are low on immunity or newborn babies. They should always be given boiled. We call it disinfected water, right? So the
Yes,
bacteria in
sir.
the water die. So he took S strain, just 1 second, let me. He took a S strain, which was heat killed. Okay. And then injected it. Now, what do you expect this time?
Will not die.
They will not die. It is the S strain.
But sir, it is heat killed.
Yes. So mice live, which means heat can inactivate the killing property of the S strain, and which is this polysaccharide mucus coat around it, because that gets degraded, damaged. But till here, everything was okay. It was as per our expectations. But this fourth thing that he did, he took S strain, which was heat killed. Okay. So heat killed S strain. What do you expect if I inject it? Mice should live, right?
Yes, sir.
But he made a cocktail with R strain. And these R strain were live. Now, heat killed S strain and live R strain, both of them alone, individually, were not lethal. So he thought, if I inject them into the mouse, nothing will happen. So he injected into mice. And what do you expect normally?
I should live, but it dies.
Yes, mice should live, but instead the mice die. Now, this is an interesting result, right? Why did the mice die? We thought that R strain is non-lethal, and we thought that S strain, heat killed, is also non-lethal. Then why did it die? Can you guess something? What do you think must have happened? If you would have gotten this result, what you would have thought?
R strain was dominant.
What then? R strain was dominant. R strain, you inject R strain, mice lives. So if R strain was dominant, then mice should live. And strains are not dominant. Genes, alleles are dominant or non-dominant. So there is no concept of dominancy in strain, because these are organisms. Bacteria is a single-celled organism. You cannot say that dogs are dominant over cats. They are two different organisms. The concept of dominancy comes in alleles. You understand? We are not talking about alleles here. We're talking about organisms. What else can you think? What must have happened?
Sir, their combination became very violent.
That we can see. The combination killed the mice. But why? This alone was not killing the mouse. This alone was not killing the mouse. Both of them together killed the mouse. What happened?
The R strain made the S strain live, come back to life.
I think that is very, very non-biological. You cannot make something come back to life once it's dead, but the other way around is possible. This S strain, which was heat killed, still had something in its cell which could be transferred to the R strain. And so the R strain became virulent because R strain was alive. You cannot resurrect something from the dead that way, but you can give some properties, some genetic advantage to the living strain. So S strain had the genetic material inside the nucleus. By heat killing it, we damaged the cell, we opened up the cell, and the cell is no longer living, no longer behaving as a living system. But whatever is present in that living system is still there. So some DNA will just leak out, will be present in the medium, and that DNA or that genetic material, I should call it genetic material because till now we do not know that if it is DNA. So that genetic material was taken up by the R strain, and the R strain became virulent. R strain is called R strain because it cannot synthesize polysaccharide chain around it, the mucus chain. And S strain can do that, which means it must be having some genes for it. Those got transferred from S to R and transformed the R into S. Now, this is just a theory. How to prove it? What can I do to prove that indeed R strain got transformed into S strain? Can you think of anything? Logic, simple logic. I am making a claim as a scientist. I am saying, I believe when I made a cocktail of S strain and R strain, the S strain was dead. The R strain was alive. Something from the S strain transferred to the R strain and transformed the R strain into S strain. So if you are another scientist who wants me to prove it, what will you suggest? Do this and prove it to me. Nothing? You can suggest nothing? So you will believe my word. If I can show you that I did not inject any live S strain, I injected this cocktail, and from an infected dead mouse, I can recover some live S strains. Will it prove my claim?
Yes, sir.
Yes, because there's only one way. The R strain got transformed into S strain because these were dead. Okay? Yes or no?
Yes, sir.
Yes, sir.
Yeah. So write down. Griffith concluded that the R strain bacteria somehow was transformed by the heat killed S strain and enabled the R strain to synthesize a smooth polysaccharide chain polysaccharide coat and become virulent, or you can say become S strain. Correct? So this is what he concluded. Now you have to prove it. So to prove, he recovered living S strain bacteria from the dead mice.
Excuse me, sir.
Yes, Arpit.
Sir, why is this all tested in a mice? No, why not in a mouse?
Why not in a?
Mouse.
Yes, the plural of mouse is mice.
Oh.
Okay.
Yes.
So it is being done in mouse only. But you cannot do something in one mouse. No, it will die. You need another one. So the moment you start using two mouse, you do not say it mouses. It's called mice. Okay. Mice is not female. If you're thinking that this is a female and mouse is a male. It's a gender neutral word, like a human. No. It can be female mouse and male mouse. Okay. So I thought you must be thinking that only.
Yes.
A mixture of heat killed S strain and live R strain was injected. So this is the proof of the principle that actually the R strain got transformed. Now, what could have been transferred from the dead S strain to the living R strain? Of course, the genetic material, right? Because that only contains the information about genes and the properties of that organism, right? The characteristics of that organism. Yes or no?
Yes, sir.
So this experiment actually told us for the first time that the transfer of genes or genetic material can happen not only across generations, so like from parents to offsprings, but also if you take it from one organism and put it in another organism, it can still bring some changes in the characteristics or the physical appearance or the physical form. Okay. But what was not clear from this experiment is, write down. What was the drawback? What was the loophole? Or no, what was the drawback? Loopholes are not the right word. What the Griffith's transforming principle could not explain. What the Griffith's transforming principle could not explain. Tell me, what do you think it could not explain? What is still missing? What were we looking? What were we searching for?
DNA, genetic material.
We were searching for the nature of genetic material. This experiment proved that yes, there is a genetic material which can be transferred, which can bring about changes in the characteristics. But this experiment did not give us any idea about what is the biochemical nature, right? So write down. Griffith's experiment did not define. Griffith's experiment did not define the biochemical nature the biochemical nature of genetic material. What is the identity? It was still the mysterious superhero and no one knew what it is. Okay. Everyone knew that it is there, which is bringing about all these changes, giving our strain the superpower to kill now like our strain and things like that. But no one knew what it is. So this biochemical characterization was done by later on 3 scientists. Okay. And they did a series of experiments, which was almost like a decade-long experiment. You know, it was done for a very long time. And there were 3 scientists, write down. The next step was biochemical characterization. Now, if you were present during Griffith's time, and you know this experiment was very, very famous, everyone heard of it. It was, you know, the topic of discussion and gossip in scientific community. Oh, Griffith showed this. So this transforming principle became very famous. So if you were there, what would you have done to figure out what is this transforming principle? Biochemical characterization. How will you characterize what is it biochemically? So inside a cell, you know, at that point of time also, people started discussing what it could be. So of course, there were some shortlists candidates. There were some favorite candidates. And the most favorite candidate to be genetic material was protein. And it is, you know, it is very obvious to think that, you know, proteins could be the genetic material because protein does every important business in the cell. Yes or no?
Yes, sir.
So proteins were already required for everything. They were also present in the membrane. So they are required for communication. They are required, they act as enzymes. They also make the cell because they are structural units. They make the cytoskeletal elements. If you have studied chapter in class 11th, the cell chapter, you know that proteins do a lot of diverse thing in the cell, right? So everyone thought that the best candidate, the best suited candidate for a genetic material is protein, but no one was sure. So how to figure it out? That inside a cell, there are, let's say, 3, 4, 5 different kind of biomolecules, which are the candidates. For example, let's say lipids, proteins, carbohydrates, nucleic acids. Let's start with these 4. These 4 are the candidates. How will you figure out which one is a genetic material and which one is not? If I ask this question, giving you an analogy from your day-to-day life, I always give this analogy in classes. So let's say you are allergic. So you eat biryani and you get an allergy. Now, biryani is a dish which has multiple components in it, you know, rice, if it's some spices, you know, other things, onion, everything is put there, plus meat, or if it is vegetarian, you have paneer. All different sorts of things are there in a biryani. It's a complex dish, right? How will you figure out which component you are allergic to?
Try removing each component.
By removing each component and then tasting the biryani?
Not everything, by one.
So let's say out of 4 components, you will remove 1 and eat the rest 3.
Yeah.
And see whether you get infected or not.
Yeah.
So don't you think you have to make a lot of permutation combinations then?
Yeah.
Suppose you removed dalchini as a component or spices as a component and you still got infection, allergy. Then next time you removed peas out of it, let's say, if it is there, and you still got the allergy. Then you removed something, but there was you will put back the other component, right? This is what you are saying, right, Fatima? So you are saying exclude one and taste the rest. What if I suggest you to do the otherwise, just the opposite? You take one component at a time and taste it independently. Will that be a better way to do it? Just taste the rice and just the peas. Yes, no?
But rice with masala, because it's not a biryani then.
Yeah, but it's not like rice with masala is causing you allergy, but rice alone and masala alone will not cause you allergy, right? If it is one component that is causing you allergy, even if you taste that component alone, it will cause you allergy, no? Nothing is changing molecularly when you are putting masala with rice. Masala is masala, rice is rice, right? This is a rule of chemistry. When you put sugar in water, if you are not allergic to sugar and to water, but can it be possible that you are allergic to sugar water? Yes, no? People, am I audible to you? So you have to be discussive. Okay, so there were three scientists whose name was Avery, MacLeod, and McCarty. So Oswald Avery, Colin MacLeod, okay, and Macklin McCarty. These three scientists did a series of experiments to figure out what is the genetic material. Okay, and before their experiment, everyone thought the most popular notion in the field was that it is protein. So what they did, their experiment was, write down the steps of their experiment. First, they purified the biochemicals. First, they did the same experiment that Griffith did, okay? And then they purified the biochemicals. And which biochemicals majorly? Which were the targets? Proteins, lipids, let's say carbs, and nucleic acids, DNA and RNA. Okay, so this technique of purifying biochemicals was developed, and they used this technique, which was not present when Griffith did the experiment. They purified the biochemicals separately from where they should purify it. Where would you like them to be purified from? From the heat-killed S strain. Okay? Because this is what we are saying, no? Is transforming the R strain. Okay? Yes, no, something? No one is speaking today. Okay. So they purified these biochemicals, and then to check one by one, which is responsible for the transformation of the R strain. Okay. So next step they have to do is to they have to culture or they have to make a cocktail of the R strain with that component, with the protein purified from the S strain, lipids or carbs or DNA or RNA. And they found, they discovered, right down, they discovered that only DNA, deoxyribonucleic acid, this chemical, only DNA from the S strain caused the R strain to become virulent. Is it clear? No, it was very clear, a conclusion. And they also tried to, just provide the proof of principle or proofread or cross-check the results by using digesting enzymes. Okay, so they used proteases. You know what are proteases? People, proteases. Proteases are protein digesting enzymes. And when they used proteases, the transformation was not affected. Okay, did not affect transformation, which tells us that protein is not responsible for transformation, because even if you degrade protein, transformation is not affected. Then they use lipases. Similarly, they used RNases. So all these three had the same result. So these are lipid digesting, enzymes, RNA digesting enzymes. So all these two also did not affect the transformation. Only when they use DNases, when they use this enzyme, which degrades DNA, they saw that transformation was inhibited. Because if you will not have DNA, if you degrade the DNA, the transformation will not take place. So this is just to prove what they claimed by another way. So do you think, are you convinced? Do you think that their conclusion that DNA was the genetic material, are you convinced? So they concluded. Yes, Fatima seems to be convinced. So they concluded that DNA is the genetic material. Okay. But their conclusion was not accepted by all the biologists. Okay, so all biologists were not convinced with the way these experiments were done, the way these purifications were done. They always, were skeptical of contaminations and things not working properly because nothing was visual. Like we could not see what is being added and whether you are 100% sure that it is DNA or not. So everyone was not convinced. Okay, though they concluded and they thought that this is a good way to do it. But since everyone was not convinced, these experiments are studied, but not regarded as the proof that DNA was the genetic material.
This is more than enough proof to say that DNA was the genetic material.
Yes, because the way we study it very clearly, so they said that when they injected DNA, transformation happened. But how can you be sure that DNA did not have any contamination of a protein in it when they were injecting it? So what they were claiming to be separately standalone DNA mixture or a protein concoction or a lipid. So the question is your purification, because these techniques were very newly developed, right? So they were not 100% convincing that there was no contamination, first of all. And second, what they are calling as DNases is not going and digesting any other component in the cell, because of which transformation is not happening. So when we read it in such plain words in the NCERT textbook, you will be convinced, right? But the way these experiments were conducted, how did you culture the bacteria in the presence of DNA, protein, and other components extracted from the S strain? So all those paradigms were not very convincing to many biologists. So in science, the good thing or the best thing is science does not believe in words regardless, from whose mouth the words are coming. Even if a big shot, even if Einstein or Stephen Hawking or Charles Darwin or anyone whose name we read in books, if they say something, the community will not just say that, okay, the god of science, the god of the field, the big people are saying this, the experts are saying this, so let's just agree to it. Science demands proof, and that proof should be without any flaws or without any loopholes. Even if there is 1% doubt that this might have gone wrong, you will not accept it as a proof. You understand? They were correct. They were not wrong, basically. They concluded way ahead of at least, I will say, 10 years, 8 to 10 years ahead of the final proof that came. So they actually saved those 8 to 10 years and they said that, okay, DNA is the genetic material. But because there was still 2-3% doubt in their experiment, so many people said that, okay, you seem very convinced, like you do not seem very convincing. So I will not take your experiments as the final proof. Maybe something better can be done to say that DNA is the genetic material. So people were not saying that you are wrong. This is, this is absurd, or this is not at all logical. Just like Fatima said, it is more than enough a proof because it sounds logical. But the way it was done, the way the cultures were handled, the way purifications were done, the way treatment with DNases, RNases were done, there were some doubts in some people's head that this could be done in a better way, or there could be a better experiment planned to show that DNA is the genetic material indeed. You understand, everyone? So this is how science operates. You need...
Yes, sir.
Yes, very good. So the final experiment that basically gave this unequivocal proof that indeed DNA is the genetic material came from these two people called Hershey and Chase, Alfred Hershey and Martha Chase. So it's called Hershey-Chase experiment. Hershey-Chase experiment. And it was done in the 50s. Okay. Now, it was early 50s, but please figure out what it was. I'm forgetting the year, but it was 51 or 2 or 3, somewhere in the early 50s. Okay. So in the early 50s, Hershey and Chase, they planned a very, very simple but very, very elegant way to prove that DNA is the genetic material. Now for that, to prove that DNA is a genetic material and not protein, you needed, okay. So I'm getting two answers in the chat that it was 1952. I hope you're sure, right? I'm just relying on you. It was 1952, okay? Fatima and Arpit gave me this answer. So if it is wrong, you know whom to blame. So Hershey and Chase, what they did, they used bacteriophages. Now, how many of you know what are bacteriophages? The name includes something like bacterio and phages. Bacterio means bacteria, right? And phages means phagy, to kill or to eat up. So what does it mean? This is something that kills the bacteria, right? Or infects the bacteria. You understand, everyone?
Yes, sir.
So bacteriophages are viruses that infect and kill bacteria. Viruses are very, very host specific. So bacteriophages do not affect humans because they only infect and kill bacteria. Now, why did they choose this as a model system to go forward? Because of this. Bacteriophages are very simple. They have a protein coat, okay, inside which there is a single genetic material, and everything else apart from the genetic material is actually made up of proteins only. They have these leg-like structures. So everything that is marked in black is proteinaceous. This is called protein coat. And what's marked in blue is the DNA, the genetic material. Now we know it's the genetic material, but earlier we knew that it is the DNA. So viruses are DNA encapsulated by a protein. Is it clear? Very
Yes.
simple structure. Now, because viruses do not have cytoplasm, other cell organelles, because viruses are not living outside the host, they only behave like living when they enter the host, where they replicate in number. Now they took bacteriophages and cultured these viruses into different medium. Okay. So first was medium 1. In this medium, they used radioactive sulfur, S35. Okay. And in second medium, they used radioactive phosphorus, P32. Now, what do you think is the point of using radioactive sulfur and radioactive phosphorus? Radioactive sulfurs will label the protein, only the protein and not the DNA. and radioactive phosphorus will label only the DNA. You know why? Fatima.
Because there is phosphorus in the DNA. There's phosphate group in the DNA which absorbs phosphorus.
Not absorbs. When these viruses will be cultured in a medium, so they will where they will replicate, okay, like in cell cultures. So to replicate, they need to make more DNA. Yes, Fatima?
Yes, sir.
And to make more DNA, they need raw materials for the DNA.
Yes, sir.
And one of the raw material for the DNA is phosphorus.
Yes.
And if the only phosphorus available is radioactive, then they will only use the radioactive phosphorus.
Yeah.
Hence, their DNA will become radioactive, right?
Yes.
Same goes for protein. They also have to make the protein coat, and for that they need the raw material. And if the raw material is radioactive, then all the protein will be radioactive, right?
Yes.
This is a brilliant way to tag protein and DNA separately without going into the problem of purifying, making it an extract and then putting it in a bacteria and then again doing all that clumsy business. So they said, we will use virus, very simple system. No one can argue that it has other things which can contaminate or things like that. It only has DNA and a protein coat. So let's do this. Let's tag and you know what is radioactive element? Do you know it already from your chemistry or physics? Sumaira, you know what is radioactive?
But they are highly reactive.
Yes, but what is radioactive? Radioactive. Sumaira, any idea? No, you have not studied about radioactive elements, radioactive isotopes of elements. So these
Emitting radiation.
Yes, they emit a radiation, right, Kulsum? Thank you, very good. And you can actually visualize if the radiated emission is in visual spectrum, you can just visualize the signal wherever these molecules or these elements are present. Okay, so it's like tagging and then figuring out, detecting the signal where the signal is coming from. Okay, everyone? And then they started with the 3 steps of their experiment. They first cultured, so write down, first thing, write down.
Sir,
Yes.
can you again explain this medium thing?
Can I again explain what?
Sir, this medium, by the separate mediums.
So they used 2 medium. In one, they used radioactive sulfur, and in the other, they used radioactive phosphorus. So these are radioactive elements. You know, they actually emit radiation. There are many there are natural radioactive elements also. These are not sulfur is not naturally radioactive, but there are some isotopes of sulfur which are radioactive, which can be made to be radioactive. You know about radium? That is where the word also comes from, radium.
Yes, sir.
Some watches, if you put the minute and the second arm of the watch,
sir, it glows.
Yes, if they have a radioactive coating, throughout the day, they absorb the energy from the sun or the light. And during the dark, they radiate that energy. So you see glowing thing, right?
Yes, sir.
So if you put a radioactive element in presence of light, it will absorb energy. And when it is dark, you will see it is emitting out energy. So you can track where that radioactive element is. You understood, everyone?
Yes, sir.
Just like in a dark, you can see time in your watch that have radium coated minute and hours arm. Okay.
Yes, sir.
So write down. Hershey and Chase cultured viruses, bacteriophage viruses. Hershey and Chase cultured bacteriophage viruses in two different medium, one containing radioactive phosphorus and the other containing radioactive sulfur. Okay. Second point, radioactive sulfur medium will allow the viruses.
Can you repeat again?
Sorry,
Can you repeat again?
which one? The last sentence or this one?
Yeah, sir, the last.
Okay. What was the last sentence, Fatima? Can you just tell me the beginning?
Hershey and Chase cultured bacteriophage viruses in two different
mediums, yes. One containing radioactive sulfur and the other containing radioactive phosphorus. Next point, yes. Radioactive sulfur will give rise to radioactive proteins.
So you were saying radioactive sulfur medium will allow the viruses?
Okay, so don't write that, write this one. Radioactive sulfur medium will give rise to radioactive protein carrying viruses. Radioactive sulfur medium will give rise to radioactive protein carrying viruses. And radioactive phosphorus medium will give rise to viruses having radioactive DNA. Okay. Is it clear? Shall I move forward?
Yes, sir.
Yes. Now, then they start.
Can you repeat the last part? Radioactive phosphorus medium will give rise to radioactive?
Will give rise to viruses having radioactive DNA.
Okay.
Because all the viruses that will grow and multiply in this culture will only use radioactive sulfur to make their DNA, right? Sorry, radioactive phosphorus to make their DNA, right?
Yes, sir.
Yeah. Now, then they allowed these viruses to infect. So the first stage was infection. They allowed, so first stage. Was infection. So they allowed, let's take two bacteria here. In this medium, let's say one bacteria. And in this medium also there are bacteria. I'm just making one representative. This is E. coli bacteria. They took E. coli. And they allowed, so in this case, let's first make the virus, how it will look like. If it is as a radioactive sulfur, then all the protein codes, right, will be labeled in red color, right? Let me, whatever is in red is representing radioactive, okay? So the virus will look something like this with the DNA, normal DNA inside, right? Yes. And in this case, where the DNA is labeled, the protein code will not be radioactive. It will be normal. with a radioactive DNA inside, correct? Okay, everyone. Now what happens is, in this case, so the structure of the virus, if you look at this, it has a head. So this is the head part. Then it has a neck region. And it has these tail-like structures or leg-like structures. So with these structures, it grips the bacterial membrane. So what it does is, the viral particles are smaller than the bacteria. They will come and sit on the bacterial surface. Okay. And the genetic material which is inside will be. Is my voice breaking for others as well?
No, it's clear.
I think Fatima, it's some issue at your end. Try to figure this out. Let me know when my voice is breaking for people. Okay. Others. So what this virus does is it sits on the membrane of the E. coli bacteria and literally drills a hole in the membrane and the cell wall and sends its genetic material inside. Okay. And the protein coat remains outside. So the virus does not enter the bacteria as a whole. It only sends the genetic material, and that is enough. This genetic material of the virus will go and incorporate itself with the genetic material of the bacteria, will get transcribed and translated to form more more viral proteins. And all these viral proteins, head, neck, and the tail proteins, they will assemble to make a new virus with the DNA inside. So from one DNA, one virus of infecting one cell, you can make millions of viral copies inside, because it uses the host machinery to replicate, to transcribe and translate. So in this case, it will look something like this, whereas in this case, the bacteriophage will come and sit on the surface. And now it has a labeled DNA, which will be, this bacteriophage will also send its genetic material inside. This is how they infect the bacterial cell. Is it clear, all of you? The first step?
Yes, sir.
Yeah. Now, as I told you, with the help of these tail-like structures, they actually bind, like kind of grip on the membrane. So the next step is important, which is called blending. Now in blending, just like you blend something in a blender, you will just agitate, mix thoroughly this the culture or the solution, and this blending will allow. So what happens in blending is, now DNA will be inside, and this protein coat, which was attached to the empty protein coat without having the genetic material inside, it will be removed from the surface. Okay. In this case also, this is removed from the surface upon blending, and the genetic material is inside, right? And then in the third step, you do the centrifugation. You know what is centrifugation, everyone? Centrifugation. Centrifugal force, centripetal force. No.
Separating particles from solution.
Sorry?
Separation of particles and solution.
So when you spin using the force of gravity at very high speed, when you spin something in a solution, in that solution, whatever is having higher density or which is heavier will settle at the bottom. And things which are lighter will actually stay in the solution only, right? So depending on what centrifugation medium you are using and at what speed you are centrifuging, you can actually separate separate two different components. Here we have to separate the bacteria with this viral protein coat, viral coat. Okay. So what happens is, in this case, let's say we are doing it in a test tube. So the heavier thing, which is the bacterial cells of the E. coli, will settle at the bottom and it will be called the sediment, right? Whereas the liquid, the protein, the bacteriophages will stay in the supernatant. Okay. The viral particles will be here. And the bacterial cells will be in sediment because they are heavier, right? Everyone understands?
Yes, sir.
Same thing will be happening on this side as well. There will be sediments, the bacterial cells. And you will have a supernatant. Okay. Now, in the first case where the culturing of the viruses were done in the presence of radioactive sulfur, the protein coat was labeled only. So we will get this radioactivity signal, this red signal in this case, in the supernatant. Okay. You understand, everyone? So the radioactive sulfur detected in the supernatant. And no radioactivity was detected in the bacterial cells. So what does it tells you? What does it tells you? That protein is surely not the genetic material because it is not getting transferred into the cell, inside the cell. Yes or no? Am I audible? Am I audible, people?
Yes, sir.
Yeah, okay. So do you understand this? That protein is not the genetic material because it is not detected in the cells. Whatever should be the genetic material should be detected inside the cell, because we know that when viruses infect the cell, they send their genetic material into the inside the cell, the bacterial cell, right? On the other hand, in the other case, the radioactive signal was detected inside the sediment in the bacterial cells where the DNA was radioactive. Okay. So here you can write radioactive phosphorus was detected in the bacterial cells. and no radioactivity in the supernatant. What does this experiment tells you? This tells that it is DNA that acts as genetic material. Because we can see the signal, we can detect the signal, radioactivity signal, and not the protein. And this experiment, the Hershey Chase experiment, is said to be, or is said to provide the unequivocal proof. Everyone will have to agree to this because this is the most logical way you can prove it without any loopholes or drawbacks. Unequivocal proof of DNA as genetic material. Is it clear, everyone?
Yes, sir.
Please draw this and finish and let me know when you're done. This is the most important part. Let me know if I have to scroll up or down. Go through everything that you have written. Take 2 minutes. Let me know if everything is clear to you. Done, everyone? Fatima, Kulsum, and Samaira?
Yes, sir.
Yes? No doubts? Are all the experiments clear? Griffith, Macri, Avery, MacLeod, McCarthy, and Hershey and Chase. These 3 experiments are clear to you? In the series in which they were done. Now, finally, we reached the point where we know now that DNA is the genetic material and everyone is okay with it. Now, later on, it was also found that many, in some viruses, RNA also acts as a genetic material. Okay. So it was figured out later on that apart from DNA, RNA also acts as a genetic material. In some viruses. Now, some examples are TMV, TMV stands for tobacco mosaic virus. Okay. And there are one type of bacteriophage which is called QB bacteriophage. So this is also a bacteriophage, but it is a RNA bacteriophage. So these two have RNA as a genetic material. Is it clear?
Yes, sir.
Now, having said that, another debate started. So DNA, RNA both can act as a genetic material. So for a molecule to be genetic material, what should be some criteria, right? Because for every job, if the job is to act as a genetic material, you need to have some skills, right? For every job, you need to have some skills. Now, what a molecule should have, which can make it fit to be a genetic material. So write down this question. This actually is asked in school examinations a lot. Write down the question. What is the criteria, or define the criteria for a molecule? Define the criteria, or they can also ask you properties or characteristics of a molecule to act as a genetic material. Now, what do you think? What should be the criteria for? Biology is logical. So if any molecule has to be chosen by the nature to act as a genetic material, what should be the criteria? For example, if you want someone to be an athlete, that person should be agile, active, not obese, right? Should have good muscle tone, stronger body build, things like that. So what should be criteria for someone, and for an organism to be able to fly? It should have wing-like structures, right, to keep it airborne. In case of birds, they have hollow bones to make them lighter and be able to fly. So what should be the correct characteristics of a genetic material? First is, it should be able to replicate itself, right? Yes or no? Because if it cannot replicate, it cannot be inherited from one cell to another when the cell divides. So DNA replicates. Now we know. So genetic material should be able to replicate. Okay. Self-replication is the property. What else do you think it should have? DNA should have? It should be a stable molecule, right? It should not easily degrade in the cell. Yes or no?
Yes, sir.
It should be chemically and structurally stable. Third, it's true that it should be stable, but what is required for cell, like natural selection and survival? What is important? Variations, right? And how do variations come? Through mutations. So it should also provide scope for slow changes like mutations, because it is very important for the evolutionary process of the organism. Yes or no? If an organism will not evolve, it will not be able to cope up with the changing climate. Yes?
Yes.
Yes. So third important thing about this molecule should be, will be that it should be able to allow, it should be able to allow slow changes, which we also call as mutations, which are essential for evolution. And last, having said all of this, if it's a genetic material, it should be able to or we can say it should be able to express itself in the form of Mendelian characters. In other words, you can say it should be capable of Mendelian inheritance. Or it should follow the laws of inheritance given by Mendel. Is it clear? So these
Yes,
four criteria should be there. Now, if you look carefully at these four criteria, all of you, protein fails at the first level itself. Proteins cannot self-replicate, right? They depend on RNA and ribosomes to be made. So they are produced in the cell. Now we know that they are how is protein synthesized? By ribosomes, right? We know that. So proteins can never be a genetic material because they do not even qualify the first criteria. Yes or no? Right? Now there are two molecules that we know act as genetic materials, which is RNA and DNA. So in the next class, we will start the discussion with DNA versus RNA. For all these criteria, for stability and everything, we will study DNA versus RNA. What is the difference in DNA and RNA with respect to stability? So write down, this is also your homework for today, that you have to read DNA versus RNA on the basis of all these four criteria. You also have to study about RNA World Hypothesis. So there is a hypothesis called RNA World Hypothesis. What is this hypothesis? Okay. And then For DNA, before you come to the next class, you will read these three things: replication, transcription for RNA, and for proteins, translation. Okay, this together is basically you have to read the central dogma. This is given in NCERT, so please go through it before you come to the next class. In the next class, I will finish DNA versus RNA, RNA World Hypothesis.